This protocol is important because it is simple to perform and within reach of any laboratory. This protocol allows the isolation of HLA or MHC type 1 or type 2 peptides from human or mouse samples. This method can address scientific questions related to all research areas involving the immune system, whether in cancer, virology, or autoimmune diseases. This protocol was built to be accessible to anyone working in a research laboratory. Just follow the steps.
Begin activating the Cepharos cyanogen bromide beads by weighing 80 milligrams per sample and transferring them to a 15 milliliter conical tube. Add 5 milliliters of 1 millimolar hydrochloric acid. Pipette up and down 5 times to facilitate resuspension of the dried beads, and then fill the conical tube with an additional 8.5 milliliters of 1 millimolar hydrochloric acid. Rotate the beads at 20 RPM for 30 minutes at room temperature using a rotator device. Centrifuge the beads at 200 times G for 2 minutes at room temperature and remove the supernatant by aspiration. Add 500 microliters of coupling buffer to the beads pellet. Then transfer the bead suspension to a new 2 milliliter centrifuge tube and set it aside. For coupling the antibodies to these beads, add 2 milligrams of the selected antibody in a new 2 milliliter microcentrifuge tube and make up the volume to 1 milliliter with a coupling buffer solution to get a final concentration of 2 milligrams per milliliter.